everyone, so our group will be presenting about the uv-vis spectroscopy. So uv-vis spectroscopy is a technique used to measure the absorption of UV and visible light, which help in identify and quantify various substances by analyzing how they absorb light at different wavelengths. So in uv-vis spectroscopy, there will be light source emitting the UV light or visible light, which will strike onto the sample cell. So the sample cell containing electron molecule will absorb the light at certain wavelength and being excited at ground state to the higher energy level, which what we call as electron excitation. So the remaining light that are not being absorbed by the electron will be passed through and detected by the detector in the machine, which will produce result that we call as transmittance.
So, how does a uv-vis spectrophotometer works? Firstly, a broad spectrum of UV radiation from a light source will strike the prism where it reaches the exit slit and where the sample is irradiated. Here, absorption occurs when an electronic transition matches the energy of a certain UV band. Then, the detector will measure how much light the sample absorbs, transmits or reflects at each wavelength. From the remaining radiation, an absorption spectrum is obtained. If you are wondering what is absorption spectrum, it is used to detect the presence of a particular substance in a sample and in many situations to quantify the amount of the substance present. Next, if you are wondering what the structure of a uv-vis spectrophotometer looks like, here is a figure for you. There are 4 main components in uv-vis spectrophotometer. The first one is the light source. It emits a broad range of electromagnetic radiation over a UV visible spectrum. The second main component is the dispersion device, which divides broadband radiation into wavelengths. There are 2 types of dispersion device, which are prism and filters such as absorptive filter and interference filter. The third main component is a sample, where the light travels through or reflects off the sample. And the last main component is the detector. It is used to measure the intensity of the reflected or transmitted radiation.
Hi, my name is Aina Shafika binti Arifin and I will be explaining regarding the sample handling of UV spectrophotometry. So for the first one, almost all UV spectra are visible in the solution phase. Quartz, plastic and glass can be used to make cells. One of the example of cells is the cuvette. And the whole 200 until 700 nanometers wavelength range is visible in quartz. Glass and plastic works well for visible spectrums. And the last one is the determination of concentration by empirical. And the next one is the graph. And as you can see in the graph, and also the formula given, which is A equal to epsilon Cl. A is absorbance, epsilon is the molar absorptivity, C is the concentration, and L is the path length centimeter. We can see that it's giving the linear effect. And looking through the relationship in Lambert-Beer's law, the longer L the sample and also the higher the concentration in the sample will be resulting in the higher the UV light will be absorbed, meaning it will be giving the linear effect of the graph. And for the molar absorptivity is determined by the orders of magnitude at which some electronic transitions absorb photons more efficiently than others. And the last one is the transmittance and absorbance, which is TT. Transmittance and absorbance A is inversely related by looking through the formula, which is A equal to negative log 10 T.
Hi, my name is Putri Nur Aida Shahida, and in this section, we will be talking about the 5 limitations of the Lambert-Beer's law. As mentioned earlier by Aina, the Lambert-Beer law tells us the amount of light absorbed based on the concentration, path length, and molar absorptivity. However, there are limitations to this law, meaning it does not always work perfectly. The first limitation is regarding high concentration. When the liquid is very concentrated, meaning that it has lots of substances in it, the molecules can start to interact with each other in ways that change how they absorb light. It's like trying to look through a really dense fog, however, it does not behave the same as a light mist. Secondly, light scattering. Liquids that are not clear or have tiny particles floating in it can scatter light. This scattering affects how much light actually gets through the liquid, making it hard to measure accurately. Thirdly is the chemical interactions. Sometimes, the substance in a liquid can change chemically when light hits it or when it interacts with other substances in the solution. This can alter how much light is absorbed. Other than that, the equipments or instruments we use in UV-vis to measure light absorption also has its limitations. For instance, if the device isn't perfectly calibrated or if the light source is not stable, our measurements can be off or we may not be able to obtain the expected results. Lastly, the non-linear behavior when using the Lambert-Beer's law. At very high or very low concentrations, the relationship between the amount of substance and the light absorbed can stop being a straight line. This non-linearity means Lambert-Beer's law doesn't always hold true.
Hi, my name is Nur Nadira binti Abdullah and I'll be explaining about the principle of spectophotometry. So the principle is when a light source shine a light through a slit, it will go through a prism and it will produce a lot of rays with different wavelength. Thus, there is a detector that will choose which wavelength to be exerted, to be shine into the detector, into the sample. So what happened at the sample is that the correct wavelength will be absorbed in the light ray will be absorbed or transmit. It depends, but the concept is the higher the concentration, the less the light will be transmitted. Thus, this is called transmittance. And when the light is being absorbed, the electron will get excited and the wavelength will go like this. Okay, that is all from me. Thank you.
Hi, my name is Nurin Shahira and I will further explain on type of uv-vis spectrophotometer. It is consists of two types, which are single beam and double beam spectrophotometer. As you can see here, in single beam system, monochromatic light from a monochromator enters the sample compartment and hits the detector directly. However, in double beam system, the monochromatic light from the monochromator is split into a sample beam and reference beam before entering the sample compartment and hitting the detector. So, the single beam configuration has a simpler design because it does not need any mechanism for splitting into sample and reference beams. I would say double beam spectrophotometer are most favorable because firstly, it can read sample and reference cells simultaneously. Secondly, it can compensate if there is any irregular and wavelength changes from radiation sources. It also allows continuous absorbance wavelength spectra recording.
Now, let's talk about the applications of the uv-vis spectrophotometry. Since this technique uses light to analyze substances by shining ultraviolet, UV, or visible VIS light through a sample, this method has many practical applications in everyday life in various fields. The first main application of uv-vis is to identify substances. In medicine, doctors and scientists use uv-vis to identify and measure different chemicals in blood, urine, and other bodily fluids. For example, it can help detect glucose levels in blood samples, which is crucial for managing diabetes. In food and beverages, this technique helps ensure the quality and safety of food and drinks by detecting contaminants and measuring nutrient contents. In forensic science, this technique is used to analyze evidence from crime scenes. It can help identify substances like drugs, poisons, and even traces of blood, which can aid in criminal investigations. In the pharmaceutical industry, this technique is essential for developing and testing new drugs. Scientists use it to measure how much of a drug is in a sample and to ensure that medications are made correctly and consistently. The uv-vis is also used to monitor water quality and the environment. The quality of water is determined by testing water samples for pollutants and harmful chemicals, which ensures that our drinking water is safe and that rivers and lakes are not polluted. It can detect things like nitrates, which come from fertilizers and can harm aquatic life. On the other hand, environmental scientists use uv-vis to monitor air and soil quality. It helps detect pollutants and toxins, ensuring that the environment is safe for humans and wildlife. Another application of uv-vis is in the study of plant pigments. Botanists and agricultural scientists use this technique to study plant pigments like chlorophyll. By understanding how plants absorb light, they can learn about plant health and growth, which is important for agriculture and food production. Last but not least, the uv-vis is applied in many industries. These industries, such as textiles and chemicals, use uv-vis to control the quality of their products. For example, it can be used to check the color consistency of fabrics or the concentration of chemicals in solutions.